Yeah, the first words, my talk is human, so there'll be a bit of humans in there. And I'm quite moved to be here, and I'm impressed by all the talks we've heard so far. I'm very moved because I was in maybe the second batch of Stéphane's PhD students, and so I was working with him in École Polytechnique back in last century on estimating deformations of stationary processes. We worked on shape-form texture, and it was really a fascinating time for me because nothing had been written in advance. I mean, each week we were discovering the topic of my PhD thesis. Basically, we just made it as we were going on, and it was really a lot of liberty. Also, there was a fun project with Jérôme. We were complaining that Stéphane was distributing to us the chapters of his book with no figures, so we were like, this should have figures. He said, okay, well, go ahead, just make the figures. So, so we ended up on this wonderful tour of making the figures reproducible, because this was a concept of Dave's, whom I said it, reproducibility, and it was early stages for reproducibility. You know, people weren't talking about it that much, but research reproducibility, and so all the figures of Stéphane's book had to be reproducible. So we were using Wavelets at the time to do all the figures, and it was really great to learn the book in that way also. So I was very privileged to be with Stéphane at that time. And in a nutshell, was things that Stéphane taught me at that time was, well, interdisciplinarity, because I had a math background, and he awakened me to computer vision, to neuroscience, and creativity, since the thesis was really very open, and also a sense of confidence and a lot of humanity, because I remember after 2 years of PhD, when I came to his office saying that I would have a maternity leave, he was like, wow, that's wonderful. And when I came back 6 months later, he knew exactly what I should be doing, and it was great help too. And it's I mean, it's a really an example. I think we should all welcome young women to have babies at any time in life, and it's so it was a great experience. And then what happened afterwards? Well, since I had been taught computer vision, I joined a group that was led by Olivier Faugeras, and there was also Rachid Derish in the group. And at the time, this group, who had been working for 10, 15 years in computer vision, were really switching, really making a huge shift, which was quite courageous on their part, to move to understand how the human brain does vision. And so I thought, yeah, this seems really fascinating. Why not do that with them? And we started thinking of modeling how we can model brain activity. And I started working more and more on inverse problems of brain activity with Alexandre Grandfort and Theo Papadopoulou. And then we worked a lot on epilepsy with the people more on the medical side, like Christian Bénard. And then I thought, like, I was working at INRIA, and at INRIA there are lots of research teams working on lots of topics, and that brain-computer interfaces is definitely something that's good to do in that place, because we can, it's very interdisciplinary, and we can join forces from lots of different teams. And so that's what we did for quite a few years, and I'm going to talk a little bit about that. So first of all, why are wavelets so important for me? Is that I'm a musician, basically, and that you know, musical notes that I perceive, they only last a while, so they're compactly supported, right? And even though I had great professors like Jean-Michel Bonny talking about harmonic analysis and Fourier, I never understood how a compactly supported function could be an infinite sum of functions that started at minus infinity and end up at plus infinity. You know, the infinite sum of representing any L2 function as an infinite sum of Fourier coefficients just didn't totally make sense for me. So it's not something that I perceive. And wavelets, everything became clear with wavelets. Following Stefan's class on wavelets, I thought, yeah, that I can deal with. I can build things with this. I'm maybe a bit of an engineer as well, and I need to be able to experiment things and feel them myself. So these images, many of you have shown them in previous talks, but they really reveal maybe why we can understand wavelets so well, and what they bring to science, because of course they're superbly mathematically well funded as well. So then, what can we do concerning these brain signals? So first of all, the type of brain signals that I got interested in are the ones that give very nice time resolution. On this diagram, you can see time resolution and space resolution of different brain imaging techniques, and MEG, EEG, magnetoencephalography, they give you like millisecond or even less time resolution, which is really nice for brain signals. Well, of course, they're not superbly spatially precise, because you can only put sensors outside the head generally, but still you can do a lot with them, and generally people use lots of repetitions of experiments that we call trials, different trials, in order to get more information. Because, for example, if you give a subject a stimulus and then you observe the response in lots of different trials, there's a lot of noise. There are lots of different sources of activity that are mixed up together. But let's say that what's related to this, the interpretation of the stimulus, may come out when you look at what's common among all the trials. So that's what's called evoked response potentials. They've been used for decades and decades, and it's quite interesting what you can get out of them. But of course, there's intertrial variability. At each trial, if you look at the motor response of a subject which is in black, and you reorder all the trials, these are different trials vertically, according to the motor response, well, you can see well, the variability that there is in this data set. And when you average the trials, of course, you lose the sense of the sharpness of each individual signal. So we worked quite a lot on this type of things, which also in which wavelets and time frequency representations were as early as the 1990s. They were already used in brain signal interpretation, because here you have different trials that are being averaged together into an evoked response potential, and in which you lose lots of the activity that is not phase locked. Okay, but if you instead of averaging the signals, you average their time frequency representations, on which you have taken the modulus to make it nonlinear, or let's say the energy. If you average time frequency representations, then you get a sense of the common oscillations that are in a signal. So this was this kind of inspired us to go to some multi-trial matching pursuits, in which you would like to find representative atoms out of a dictionary, a pre-constructed dictionary in this work. You would like to find which is the most representative atom of each trial. So M are the different trials, and you would like to find to adapt your atom to adapt to each trial while retaining information from the whole data sets. So how to get information from the whole data set? It can be via averaging all your across trials, but this in fact will give you just the same as if you were using the average signal to start with, or else, as Rémi suggested, using an average energy approach, which there is different from averaging the signals across trials. Actually, when Rémi did it, this is more for multi-channel data rather than multi-trial data. In fact, so both approaches, actually, when you use both types of approaches, you still get the same dictionary atom picked across trials. It's only the amplitude and maybe the phase that are going to change, but not the actual atom. So we had another point of view on that, is to do some clustering, to do a matching decomposition on a dictionary for each trial, and to cluster the peaks of this multi-resolution decomposition, to cluster the peaks, to find in each trial the best consensus atom, and then to find the consensus atom out of all the trials, and then find in each trial the atom that is closest to the consensus. And here we can account for jitter, for variability of latency, for variability of other features, because we use dictionaries in which there was different lengths of atoms, different frequencies, different latencies, etc. So using this type of analysis on, well, here's epileptic data where electrodes are inside the brain. Why this type of data and not just EEG? Actually, it's because the signal-to-noise ratio is better. And we had trouble actually using it with actual EEG data where the signal-to-noise ratio is very, very low. But with intracerebral EEG data, it was very interesting for the neuroscientists who take advantage of electrodes being into subjects' brains to do cognitive experiments, to learn more about the brain. So it's interesting because it can give a better idea of the length of brain phenomena, and just more information can be achieved. I'm presenting things as if things were almost always the same when the brain reacts to a stimulus. But actually, the brain reaction to stimuli is very dependent on the brain state of the person. And the rest of my talk is about attention, like neural correlates of attention. Here, I'm presenting 3 types of experiments. You can present somebody with just a target. For example, you're at a red light, and you're waiting for the green light. And when the green light comes, that's your target. So there, definitely you have a P300 response. P300, 300 milliseconds after the event, there's a huge surge of activity in your brain. It's a good signal-to-noise ratio that you can see easily from outside. And everybody gets that at a red light unless you're asleep. Then if you're not just attending a single type of stimulus, but like there are many common stimuli and sometimes a target stimulus to which you should attend, that's called the oddball paradigm. And when the target stimulus is there, your response is higher than when it's just the standard stimulus. So you can see that already the response is modulated by your attention and the sense that you're putting to the stimuli. So for example, if it's music notes, one like high notes in the middle of lower notes, and that's kind of random, well, you get this stronger P300 response. And then you can even decompose the P300 response into different components. If you have a target like before, but sometimes a distractor, then the distractor is going to give you a novelty response, like, ooh, this is new. So an extra response that comes even earlier than the other. So you see that all these responses are actually modulated by attention, and it can be really interesting to try to learn about human attention using EEG. Also, it can be there are some responses that are even not attentional at all, like the mismatch negativity, which is pre-attentional, which occurs even when you're sleeping. If you're listening to beeps while sleeping, if there's a beep, well, you can see in your brain the response to these beeps, even if you're not attending to them. And these are used to see if people are in a deep vegetative states, etc. If they're in a coma, it can be used to see if there's a chance that they can come out of a coma. And then also to show you that you can be really perceiving things differently according to attention, I just want to make a little experiment with a metronome. So it's this tap. So it's just a regular tap. But if you like, because boring, you can decide that's not a regular tap, it's a waltz. So you can just decide, it's the same, you can just decide that every third beat is going to be like stronger, and maybe we can dance. Okay, I guess you can feel the meter if you want. If you want, you can decide that it's just a very fast military march, and it's just one every 2 beats that's accentuated. So if you're doing the experiment, you're feeling something different, but it's always the same stimulus. So it's just to show you that your attention is playing tricks. It's not just tricks that you feel, but we can even measure it. If we measure your EEG while you're doing these exercises, we can actually see in the brain that the brain is responding differently to the same stimulus just because there's attention there. So I got interested in brain-computer interfaces because it's a kind of challenging. It's really challenging to be able to interpret the brain activity while it's going on, right? And what can we do with it? Because the point wasn't just to do like papers, but to try to help people out with that, to try to help people who are disabled. So there's a kind of common paradigm in brain-computer interfaces. It's called the P300 speller. So the P300, you know, I just explained what it was. And the speller is that if somebody wants to type, like to select a letter, they're going to look at the letter that they want to type. And just like the green light effect, when this letter is going to flash, you get a P300. And so by looking at the P300 in the people's brains, you can guess what letter they want to type. That's the idea. And so we were contacted by Nice Hospital one day with an occupational therapist. Her job is to equip people with devices to help them continue their life when they're severely disabled. And so she worked with amyotrophic lateral sclerosis patients as Charcot's disease or Lou Gehrig disease. And once she was really stuck with this lady, actually, because this lady couldn't move her muscles anymore, and so she had trouble still using her computer. As long as you can use a computer, you can still do a lot of things. And she told us, okay, please find a way. And so we came with we put together a software to interpret P300 spellers, and the first word she wrote immediately, this person understood how to use it and wrote "J'adore," I love it, and we were quite impressed because we had 20 patients, all ages up to 93 years old. They each came for 3 sessions. Each time they were able to use it immediately. The 93 years old had never used a mouse in her life, but this, easy. I mean, it's just attending to letters that you want to type. Only it's very slow. I can give you just an example, an idea of how slow it is. So this lady has still some motor capacity. She usually writes on a tablet, and here she's trying to type a word that she thinks on her own. So it needs a few flashes because several letters are flashing at the same time. So you need at least 2 flashes to discriminate which of the letters it is that the person is attending to. And so here she's writing the word rêver, I think. And it's funny to see the family in the room saying, "But isn't this telepathy? How are you doing it?" So it's quite interesting to work in that field because you get to explain to people what is machine learning, what are the limits, what it can do, what it can't do, etc. So the problem with this type of thing is that it needs to be calibrated because each person has a P300 that's a different shape, etc. So it needs to be calibrated for each person who has to type her name or something like that at the beginning, some word that we know, so that we can get information out of the particular signals. So we did a lot of, well, maybe not enough, but we worked on also covariate shift because to be able to auto-calibrate so that people can use pre-calibrated data, and we would move their P300 to a pre-learned database. So here you have blue and red are one subject, target and non-target responses. Green and orange are another subject, and you see that they're very different, and it would be hard to just segregate them if we mix the two together. And using optimal transports, inspired by Courty and Flammary, we were able to do some domain adaptation. But it's not online yet. We don't have the software that actually does it. Nevertheless, a patient in Chambéry heard of our experiment in Nice and wrote an email to us saying, I want to be part of your study group. I want to test this because I use my eye blinks, I use stuff like that, but one day it might not work, so I want to have another way of communicating. So we told him, we can't come to you, but we'll send you the software. And we tried to make the software as plug and play as possible, like adaptable to all screens. It's a little technological, but it's worth it because in the end, this guy, Damien Perrier, who is in a hospital in Chambéry with the help of a friend who came every Friday to try to make him test it. Actually, he was able to use it, and what's very impressive is that he was using very low quality EEG equipment. He bought a $200 Emotive Epoch. It's just it uses salt water and not gel. It's very it's hard to get good contact on the head, etc. And so it's very slow. It's terribly slow because since the signal-to-noise ratio is low, it needs even more repetitions of the letter to get one letter right. So here he doesn't have the first letter yet, right? So still it's coming, and that's it. It's an M. It's slow because the data quality is low. And here it's being fast-forward because it's just so slow. But there's interest. It gives us a drive to continue working and stuff like that because you feel that people really want it. So just to continue on attention, but on another topic, I got interested in auditory attention detection because, as I said, I really like music, and it's interesting to see how the brain processes music as well. And so here you can see that if you're listening to speech, somebody talking, and you look at the speech envelope in black, it's quite well correlated to the neural activity on the auditory cortex. But here it's intracerebral. Here again it's electrocorticography, good signal-to-noise ratio, and we can see that we can decode speech envelopes from electrocorticography. So we got interested with my last PhD student, Joanne Belot, on auditory scene analysis. In a way, the cocktail party effect, how the brain does it. Well, at least can we use brain activity to find out what somebody is listening to when there are several streams of attention going on together. So he had two experiments. He was a cognitive science student, actually. The first time I had a student from cognitive science coming to me to do like experimental work and decoding. So there was a monodic condition, and it was the first time this was done in music, because people had worked on such things in speech before, but this time we were listening to musical pieces, so piano pieces like Mozart or Beethoven or Haydn, or little excerpts of actual piano. And in the monodic condition, is the same piece on both ears. And in the dichotic condition, you're listening to Mozart on the left ear and Beethoven on the right ear. And we asked the subject to attend one or the other, and we try to see if in the EEG activity we can see what they're listening to. And it was co-funded by a cochlear implant company, because in cochlear implants you kind of amplify everything the same way, and it's even more interesting if, according to what you're attending to, you could be able to amplify different things. So what it turns out is that in monodic performance, like we had 16, I think, no, 8 pieces of music, and the classification performance was over 75%, which means that we were able to say with 75% accuracy which piece out of the 8 it was. Okay, so it's just it's not really very useful in real life. In the dichotic conditions, much more useful in real life. You would have 2 streams, and you would like to know from the EEG activity which of the 2 the person is attending to. Here, the performance was a bit above chance. Well, above chance for some of the participants, and much lower than chance for others. And so here we have another individual variety of performance. So my student, being a cognitive science student, he started more looking at the cognitive reasons why there might be differences in performance. And it turns out that attentional inhibition, so attentional inhibition is very important to stay concentrated and stuff. And he measured it with the auditory Stroop test, in which you have to respond to high, like high or low, except that sometimes the person says high or low. So you have to respond to high with your right hand, but sometimes high is said in a low tone. So this is a way to measure attentional inhibition. And he found out that among all the things he tested, there was musical ability, inhibition, working memory, lots of different cognitive traits. This was the one that could explain part of the performance, attentional inhibition. So now he's working on could maybe cognitive abilities be trained. People who wear cochlear implants, maybe if we train them to have better attentional inhibition, then they might be able, even without enhancing it neurally, they might be able to better hear. So just to conclude, some food for thought. What's nice with EEG and MEG data is that it's scarce data. It's not big data. So you can't do much with deep learning yet. So what's nice, but it's hard also, because you really need to spend a lot of time to get this data from cooperative subjects. So the data is expensive in a way in time to get. There are also lots of sensor issues. As I said, this person who was using low quality equipment, he had no low quality data, right? So that's also a bit of a problem, and that's where the research needs to go. I think the most and the fastest is in sensor improvement. Still, it's super interesting to think whether machine perception can help human perception, can help human communication, enhance human abilities, of course, being careful of all the ethical issues, but also could machines help you perceive your own brain activity, which is totally, you're insensitive to your own brain activity, right? You can never feel it, but for epileptic people, for example, who have really disorders that are very measurable from EEG, could we help them perceive something in their body that's related to their brain? And that's something we're trying to do with neurofeedback for epilepsy. And I want to say again, happy birthday to Stéphane. And just sharing a memory that I had, I don't know if you remember when Gabriel was giving his first talk in Nice, ICCV 2003. You came with Milena to my house, and I was keeping Milena where you were attending Gabriel's talk. So, and that was, well, I was expecting another. So there are lots of fun memories.
I wonder, can you use these tools in order to evaluate attention disorders, ADHD?
Oh yes, definitely. we've worked with, I mentioned Jérémy Matou, he uses a P300 in a kind of game, I don't know how it's called in English, but it's called Puissance 4 in French. Puissance 4 is like you select a column in which you want to drop a ball, and you have to make a pattern with the balls. You know how it's called, cross 4 maybe. And so he uses it with ADHD children because it's a game. He measures their P300, which is related to attention also, and you're able to get children to be better concentrated, because you can see whether the game the way you're making them play the game has a relation with their activity. So yeah, definitely you can use it for that. I also worked on visual attention using oscillatory activity of the brain to detect in which part of the visual fields somebody could react fast if you throw him a ball, for example. So we have a work there where you can increase target response by giving the target on the right part of the visual fields. So it's kind of eerie, but that's more for enhancing performance, getting people to be more productive. Yeah.
Yes, Stéphane wants to say something.
Yeah. Well, thanks very much for this fascinating talk. What you showed is that one of the big difficulties, in some sense, the rate of information. It's very slow. And when we are thinking of reading in the brain, we're thinking of someone who thinks about something, and you can recognize it. So, for example, you think about an A, a B, a D, and you recognize it. How far are we, or what is the big, is there any hope to do that? Or the signals are much too noisy to be able to directly get access?
Well, what's good is that I think it would never happen without you knowing about it, because to be able to have sensors that would be able to do that, you have to have sensors that are really close to the brain, I think. So people do try decoding thoughts by placing sensors within the brain, right? But so you have to be really.
But outside the brain, you couldn't get a letter, for example, thinking about an A.
No, never. But inside the brain, yes, because when you imagine talking, if you just imagine that, there's your sensory motor, even the motor and auditory cortices that are really working together to produce this impression of, and so even without speaking, you can decode speech and inner speech. I think if you have enough sensors, so there are not that many people that are willing to open their brain to such experiments. So in a way, it's nice that our thoughts kept nice and secret, but it could happen. Of course, there are also other ways, like you can also decode from your throat. When you're imagining talking, your throat also makes faint movements, which can also be used and are used in the army or such for decoding speech.
Right, so thanks very much.